Hello everyone and welcome to this class. In this class we are going to study about enzymes. I am going to teach you in a skip way such that you can easily prepare for exam. Alright, let's not waste time and continue. So enzymes are the biocatalysts that enhance the rate of biological chemical reactions without themselves being changed in the process. So why I have written here biological chemical reactions? Basically they are biochemical reactions which occur in our body. Okay, so these enzymes are found in the body of an organism, whether it is multicellular or unicellular, and these enzymes can enhance the rate of reaction. Okay, like if product A plus B will convert to C in, let us say, 1 year, then if the enzyme is there, then it will take only 1 month. Okay, so you can see that it will decrease the rate of reaction time of the reaction significantly. Okay, so some unique features of the enzymes are: first, it exhibit high catalytic power, right? So this here is the example of its high catalytic power. Okay, so what we can say, like, there are a lot of substrates in the environment. Okay, so only one enzyme is needed for all these substrate to change into product. All right, now the second point is enzymes are highly specific. Okay, let us say this enzyme work on this substrate 1. Okay, this enzyme 1 works on substrate 1, then it will only convert substrate 1 into product 1. It will not act on substrate 2, so that product 2 will not form. Okay, so this is high specificity for the enzyme. Now most enzymes are proteins. Why I am saying this? Yes, it is very true that most enzymes are proteins, but there are some enzymes like ribozyme. Okay, so the ribozymes are not protein. Ribozymes. Ribozymes are actually RNA enzymes. Okay, that's why most of them are proteins, but not all. Okay, and the fourth point is enzymes do not change equilibrium, but it changes the rate or at which the equilibrium is attained. Okay, let us say A plus B produce C. Okay, and when equilibrium is attained, then you can say this is a substrate. Okay, so we have 40% concentration of substrate, which is converted to product, that is C, product, and the C is 60% at that time. If we introduce enzyme, this equilibrium will not change. Okay, it will not happen that this percentage will decrease and this will increase. But what happens is, like A plus B, if needs large amount of time to convert to product, and then the equilibrium is attained, then this time will be reduced such that, let us say, the equilibrium is attained at 10 minutes. Now after the enzyme concentration, this equilibrium will be attained in 1 minute only. Now let us move how the so how the enzyme is divided. Not actually divided, but rather I can say how it is made. Yes, that will be a good term. All right, so basically enzyme, first of all, as I've said, can be of two types. It can be of RNA and can be of protein type. So 99% of the enzymes are of protein in nature, while 1% is RNA nature. And the example for RNA enzyme is ribozyme. Okay, so enzyme here, if they are protein, then they can be further of two types. First is simple enzyme. Okay, so the simple enzyme only has protein component, and this protein component can change substrate into products, while the second type is conjugate enzyme. Okay, so this conjugate enzyme contains a protein part and a cofactor. Okay, so the protein part is just simple protein part, which is called apoenzyme. Apoenzyme, oh, have I written here? No. Okay, so this protein part is apoenzyme, and the cofactor is called, like, cofactor which is non-protein part. Here I have written is apoenzyme is the protein part and the cofactor is just cofactor, which is non-proteinaceous part, and they together, when combined, they form holoenzyme, and this holoenzyme is functional, while only if there is apoenzyme, then it is non-functional. All right, And this conjugate enzyme, which is non-protein part, uh, cofactor, which is non-protein part, can be further of two types. First, it can be a metal ion, okay, and another it can be a coenzyme. So, coenzyme, metal ion is basically metals, okay. So, if it is tightly bound, then it is called metalloenzyme, and if it is weakly bound, then it is called metal activated enzyme, okay. So, now let us move to the naming of the enzymes, okay. So, naming of the enzyme is like this enzyme is divided into six classes. The first one of them is oxidoreductase, oxidoreductase, and the number one is given to it. Then the second is transferase, number two. Number three is hydrolase. Number fourth is lyase, lyase. Number fifth is isomerase, and number sixth is ligase. Okay, so numbers are given like this. You have to remember these numbers, like oxidoreductase is the first class, this is the second class transferase, third class is hydrolase, fourth class is lyase, fifth class is isomerase, and sixth class is ligase. Sorry. Okay, so what happens is, let us suppose we have an enzyme, um, which is suppose is of class 1, which is oxidoreductase, and the second digit of that enzyme, so we first have to like name of that enzyme will be 1 ratio, like second digit is depends on the donor, like if the enzyme has higher donor, then it is called, it is given number 1. Let us say it has higher donor, so first number second digit is given to this, okay. Now the third digit, which is acceptor, let us say it also has classes 3 4, okay. Now the its acceptor value is a little lower compared to other, so we have to again write ratio, and third is number 3 is given, and the fourth digit is special digit, which means it is the order of the enzyme which is added to our dictionary. Like let us say we have A enzyme and B enzyme. Now A enzyme is discovered first from the oxidoreductase family, and B enzyme is added later to the dictionary. So one number is given to A and second number is given to B. So the if we are talking about the second enzyme, then number 2 will be placed here. So the name of the enzyme will be 1 is to 1 is to 3 is to 2. Okay, that's how the naming of the enzyme is done. Now there are two main theories of how enzyme operate. The first is lock and key theory, which is given by Emil Fischer in 1894. So as you know that we have a lock, okay. Now you know that there is always a key that can open the lock, alright. So this key will go and get, you know, the lock get locked in the chamber, and that can open the lock, alright. So this is how enzyme work. Like if this is enzyme and this is substrate, okay, then this enzyme can go and bind to the specific substrate and a complex will be formed. So this enzyme substrate complex will be formed, then only substrate can be converted into product. This is substrate here, okay? Enzyme substrate complex. So this is what is said in the lock and key hypothesis, while the second is induced fit theory. So induced fit theory is like this is enzyme and let us say this is the shape of substrate. Okay, now what happens is enzyme goes to the substrate and uh, some conformational changes occur in that enzyme, okay, such that it can fit to the substrate. All right, and that's how enzyme and substrate complex form, which then convert substrate into product. So these two are the major theories which we study in the enzyme, yeah, which we study for the enzymes. Now, the main thing about the enzyme is its ability to lower the activation energy. Okay, so let us say this is the reaction process without enzyme. So what happen here is like this is the substrate concentration at the initial. When a sufficient energy is given to this substrate, then only it is converted into product, or you can say break down into product. Now let us say this activation energy which is needed for the substrate to convert into EA, let us say this is 100. Okay, so what we do is we when add enzyme into it, enzyme add to this substrate, I mean this reaction. So what happens is it lowers the activation energy. All right, so as you can see here, see this here, the substrate needs less energy compared to the earlier reaction. Now here let us say EA's value will be 10 and this will convert into product. Okay, so what happens here is, let me change the pen first. Okay, so substrate plus product need EA is equal to 100 to convert this, sorry, substrate and nothing, only substrate to product, need activation energy of 100 to convert the substrate into product. While when substrate plus enzyme is there, then it will form enzyme substrate complex. Now this enzyme and substrate will have some bonds in between them. I'm drawing it like this, okay? These are bonds. So what happens is when these bonds are broken, so it will release some energy, okay? So this energy, it lowers this activation energy such that enzyme can easily be converted into products. Okay, and after that enzyme comes out of the substrate, detach itself. Okay, detach detach itself from the substrate. That's how enzyme usually work. So that's it for this class. If you have any doubt, you can always put the question in comment. Thank you very much and see you soon.